Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled, "The Race for Children Act Will Change the Landscape for Pediatric Cancer Research. Are you ready for August 2020? My name is Candace, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. Now I'd like to thank Medpace, who develops the content for this presentation. Medpace is a scientifically driven, global, full-service clinical contract research organization providing phase 1 through 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and disciplined operating approach that leverages local, regulatory, and deep therapeutic expertise across all major areas, including oncology, cardiology, metabolic disease, endocrinology, central nervous system, and antiviral and anti-infective. Headquartered in Cincinnati, Ohio, Medpace employs approximately 3,200 people across 37 countries. Now I'd like to introduce our speakers for today's event. David Horton is a subject matter expert in pharmaceutical drug development and non-clinical regulatory strategy with over 16 years of research and drug development experience in academic, government, pharmaceutical industry, and CRO settings. He has strategic development experience with virtually all therapeutic modalities across multiple therapeutic areas and all stages of drug development. In previous roles, he contributed to the design, execution, and oversight of CNS, cardiovascular, and in vitro safety pharmacology studies. He's been responsible for advising on efficacy strategy and translational pharmacology in the development of addiction pharmacotherapies in the neuroscience portfolio. He has also been involved in all aspects of non-clinical drug development, including safety pharmacology, as well as general toxicology, genotoxicity, immunotoxicology and development, and reproductive toxicology. Our next speaker is Peggy Kaiser, who has over 30 years of experience in pediatric hematology, oncology nursing, and pediatric clinical research. Prior to coming to Medpace, she was a nurse practitioner at Cincinnati Children's Hospital Medical Center in the Cancer and Blood Disease Institute. She served as Senior Clinical Research Manager of the Institute's Clinical Research Core. Her expertise includes executing complex pediatric hematology-oncology clinical trials involving many different types of investigational products, including gene and cellular therapies. And now, without further ado, I'd like to hand the mic and the presentation over to our speakers, who may begin when they're ready.
Okay, thank you very much, Candace. On behalf of myself, Peggy, and Medpace, we are very much excited to have the opportunity to speak to you today regarding pediatric oncology drug development. What I'd like to do today is really start off by just giving a background for pediatric drug development in terms of the regulatory framework. I'll then move into the Race for Children Act, highlighting some of the key challenges that are encountered in pediatric oncology, and then sort of discuss some of the purpose of the legislation, some of the key elements. One of the big components of this is the substantially relevant molecular target list, so we'll go into some of that. And finally, we'll wrap up the first part of the talk on key implications and considerations for how the Race for Children Act affects your drug development program. Next, I'll turn it over to Peggy. Peggy will discuss operational considerations for running pediatric oncology clinical trials. So she'll go into some of the key aspects of this, such as research protections, some study design considerations, what types of strategies you can employ for site selection, tips and tricks for recruiting and retaining patients, and then speak a little bit about assessments that are unique to children. So with that, I'll start off with the regulatory part of the talk and describe the regulatory framework for pediatric oncology as well as the Race for Children Act. So one of the key issues I think that is facing pediatric drug development is that while there's a considerable subset of the population, historically there have only been a small proportion of drugs that are specifically labeled for pediatric use. And what this really led to was widespread off-label prescribing by clinicians out of necessity. So they're faced with a conundrum of, do I treat the pediatric patient off-label with very little information, or do I withhold or delay treatment, since it's not approved for in that population? So really, it's honestly the off-label prescribing is dependent upon the best effort or the judgment of the clinician. So again, they're basing these treatment decisions off of little to no clinically meaningful data in this patient population from a safety or an efficacy perspective. And so with that in mind, treatment that they could be prescribing could be ineffective due to the fact that they've got the wrong dose, the wrong dosing regimen, or the wrong formulation for their pediatric population. And, the FDA and other regulators and, the field in general have sort of really felt that pediatric populations should have access to therapies that have underwent the same rigor and that have been properly evaluated for pediatric use. So two of the fundamental components of pediatric regulation are really the BPCA, which is the Best Pharmaceuticals for Children Act, and PREA, which is the Pediatric Research Equity Act. The FDA tends to describe these as the carrot and the stick. So the carrot would be the BPCA, and that was originally introduced back in 1997 as part of the FDA Modernization Act, or FDAMA. And really what that did was the carrot was the introduction of the 6-month extension of market exclusivity. This had a natural sunset date of January 1st, 2002, but in 2002, this BPCA was passed, which amended and reauthorized the exclusivity provision in FDAMA. And so how this typically works is that the FDA will issue a written request to the sponsor to conduct studies that for molecules that would be relevant for pediatric investigation. The studies are then performed to the level that is specified in the written request by the sponsor, and then exclusivity is awarded irrespective of the outcome. One key point of this is that this applies to all potential indications for the active ingredient, not just the adult indication being pursued. So therefore, sometimes some of the focus on developing treatments for pediatric use is lost, given that this applies to all indications being pursued for that molecule. Another issue with BPCA is that oftentimes these studies are completed late in the life cycle, as exclusivity is running out, and therefore you don't get that proactive approach to understanding the effects in peds, and so therefore, again, you've got that off-label use of drugs in the populations for many years without data. The process for obtaining and satisfying these written requests from the FDA can be somewhat challenging. So again, BPCA is a voluntary process. So if BPCA would be the carrot, PREA would really be the stick that the legislators use to mandate pediatric research. So PREA got its origins in 1998 with the pediatric rule, and the pediatric rule really came about to allow the FDA to mandate pediatric assessment of new indications and new development pathways. In 2002, it was invalidated by federal court as they stated that the FDA overstepped their bounds in terms of its authority to require these studies. But in 2003, PREA was passed, and that established FDA authority to mandate pediatric studies. So again, as BPCA is voluntary, PREA is mandatory. And really what PREA is founded in is the pediatric study plan. And these are required for all CDER and CBER applications within 60 days from the end of phase 2 meeting. If your program does not have an end of phase 2 meeting, then it needs to be done within 210 days of marketing authorization. And again, This applies to new active ingredients, indications, dosage forms, new dosing regimens, new routes of administration, except where that's been waived or deferred. And typically, generic and orphan drug products have been exempt, which I'll touch on in a little bit. But PREA and BPCA were both reauthorized in 2007, and then FDASIA in 2012 made them permanent. So as a result of these pediatric legislation initiatives, there have been quite a bit of activity in the pediatric development space over the years. And so this figure is from the literature, and it shows since the passage of the introduction of FDAMA and the pediatric rule, you can see that there's been an increase in the number of labeling changes over the past 2 decades. And so in fact, this legislation has resulted in more than 770 labeling changes for pediatric use in the past 2 decades. And a lot of this has really been driven by PREA, as indicated in the blue bars in the figure. And so it's, while pediatric legislation has been reasonably effective in all, many of the therapeutic areas, the results in pediatric oncology have been less obvious. And so this is a really good slide that I pulled from an FDA presentation in April of this year, and I think it really shows sort of some of the challenges that you see in the development of pediatric oncology therapies. And so, again, PREA effectively has little relevance to the pediatric cancer drug development. So this focuses more on FDAMA and the BPCA. And so, this is showing the approvals and label changes for pediatric oncology products, both pre and post FDAMA. So prior to FDAMA, only 15 label changes occurred in approximately a 45-year span. I think following introduction of the exclusivity, we saw an increase with about 26 different label changes or approvals happening in the past since 1997. A lot of these, there's a subset of these are just more supportive and less curative or more treatment oriented. So why is pediatric oncology development sort of an afterthought, if you will? And I think a lot of that is for a variety of reasons. I think adult oncology therapies are typically used to treat cancers that are typically rare or they don't occur in children. As Peggy will get into a little bit later, development programs for pediatric oncology treatment can be risky and can be quite challenging. And so therefore, a lot of sponsors view these as unprofitable. And a lot of that is coming from the market. There's a smaller number of cancer cases for children compared to adults, and couple that with the challenging study considerations, and you see some difficulty in pursuing pediatric oncology treatment. Also, with adverse events, the adverse event profile could look completely different in the pediatric population. It could be adverse events that are unique to children as compared to the adult, or even the adverse events could be amplified in the pediatric population relative to the adult. Nonetheless, I think there's been a wealth of research recently that shows that there are still drugs for adults that haven't been tested or approved in children, despite a lot of these targets that could have effects in pediatric cancers. And I think that's really come about lately because of the advancement of precision clinical medicine, or precision cancer medicine, sorry. And really, a lot of the advancements that have happened recently to increase our understanding of cancer etiology and sort of the underlying genomic factors that are involved in some of these cancers. So this brought about an initiative called the Race for Children Act. And the RACE stands for Research to Accelerate Cures and Equity for Children. And it was incorporated as Title V of the FDA Reauthorization Act, or FDARA, and that was enacted in August 18 of 2017. It's important to note that the Race for Children Act does not change BPCA, but rather it amends PREA. And really, the focus of this is to require assessment of drugs and biologics that are intended for the treatment of adult cancers and directed at molecular targets that are substantially relevant to the growth or progression of a pediatric cancer. And I'll dissect this statement a little bit further in the upcoming slides, but this is the main focus of the Race for Children Act. And effectively, it eliminates the orphan exemption for pediatric studies that are for relevant molecular targets. It's no longer just an indication basis. It's based on the molecular targets themselves. So the timeline for enacting this, It does come into effect August the 18th of 2020, so sponsors are required to submit PSPs with these considerations in mind for marketing applications that are submitted after August 18th of 2020, unless obviously the PREA requirement is waived. So, so what are these pediatric assessments? So, the FDA sort of looks at the requirement for pediatric assessments to generate clinically meaningful study data to inform the pediatric label. So are you taking into consideration all of your preclinical data to inform on your study design? Do you have the appropriate formulation developed for pediatric use? Do you have the appropriate starting dose? Are you using the extrapolation approaches that laid out in the guidance to have the best starting dose and dosing regimens? Are you considering all the variability that you have in sort of the physiological characteristics between adults and children? Do you have the right pharmacodynamic endpoints, or you've got the right biomarkers selected? And again, finally, and very importantly, are you leveraging the adult safety and efficacy data to inform on the trial design? The FDA, one thing that's been apparent is that they've encouraged early meeting with sponsors to really develop the right approach and strategy to develop these pediatric assessments and what type of information they're looking in their PSP. So I'm not going to spend a lot of time on this because Peggy's going to go into a lot of these details, but I did want to highlight some of the main guidances from ICH and FDA that are incredibly relevant to the considerations for study design. So ICH E11 is the key guidance, and really that has consideration for clinicians across all therapeutic areas. It was recently updated in April of last year, and it has a lot of guidance to sponsors regarding age classification, ethical considerations, study design, formulation challenges, etc. And then there were two recent guidances that came out in March of this year that are more specific to pediatric oncology trials, specifically around including adolescent patients as well as age requirements and eligibility. So Peggy will go into a little bit more detail of some of these later, but I did want to mention these as being important for study design. So the key focus, obviously, of RACE is to require assessments for drugs that go after substantially relevant molecular targets. And so, one of the key things that that was key in the initiative of implementing this was to understand what are those relevant molecular targets. And so, the FDA developed the molecular target list with input from the National Cancer Institute and the pediatric research community. They based the inclusion of targets on this list upon published peer-reviewed literature, abstracts, and public databases. And so the lists are published on the Oncology Center of Excellence Pediatric Oncology Program. And these target lists include either molecular targets where there's evidence that they are relevant to the growth or progression of pediatric cancer, or they're targets that are not involved in pediatric cancer, and therefore that these assessments would be waived. So really the inclusion of targets on this list comes down to the definition of molecular target. And so, a lot of this work was pioneered by Greg Riemann from the FDA, and he sort of felt that the molecular target definition was, that these were molecules in human cells that are associated with a particular disease process in cancer. And again, this could be cancer progression or cancer resistance, for instance. And these are targets that can be interacted with via a small molecule biotherapeutic or other therapy to affect that disease process. And so the FDA breaks down the substantially relevant target list into a few categories, which includes gene abnormality, cancer cell lineage, tumor microenvironment or immunotherapy, or another category. And I'll get into just a little bit of detail on the next few slides on these, but that's the breakdown that they currently have. And then they've also got the list which is not relevant and are up for automatic waivers. So the FDA and the NCI really looked at a framework to classify these targets using a weight of evidence. So what was the presence of the target in the pediatric cancer? What was the function or the mechanism of action of that target or downstream signaling pathways of that target? What kind of non-clinical or clinical evidence was apparent for that role of that target in oncology? What kind of biomarkers? And then where's the location of the target on the tumor cells or on the cells? So next I'll get into a little bit about the targets. I'm not going to spend a lot of time, and I'm certainly not going to go through these in great detail, but I did want to highlight a couple key points. So the first category of targets are the gene abnormality. And again, these lists are taken directly from the FDA website. These are targets that are present in a critical biological pathway, or that they exist a direct relationship of this gene or target with cancer. And as I said, these are on the website, but you can see a couple, big players in the oncology space, such as ALK, EGFR, there's the Janus kinases, MYC, Notch, PI3 kinase, RAS, TYK2, some of the ones that you typically would see. And then, the gene abnormality targets are the ones that have been, probably have the most validated targets or the most credentialed targets through the literature. Next, we sort of have the cell lineage target. And again, these are targets that are related to the cell lineage of cancer. And a lot of these could be, what you see here are targets that are very popular with some of the immunotherapeutics. So you're looking at your CD19, so for your CD19 CAR T therapies, your monoclonal antibodies, ADCs, etc. The next grouping is the sort of the tumor microenvironment and immunotherapy. These are targets that really affect the microenvironment of the tumor and involved in supporting sort of the cancer growth and progression. A big category here are your checkpoint inhibitors. And these are the ones that, obviously have quite a bit of activity within the oncology space. And finally, there's another category, which is sort of, your miscellaneous bucket. And this list of targets is pretty lengthy, but these include targets that could interfere with cell growth, differentiation, development. As you can see, a lot of these targets are currently under consideration for therapies within the oncology space. And then with all of those large lists, here's the very small list, which includes targets that are not relevant for pediatric oncology. And these are the targets that would be open for automatic waivers for not having to conduct pediatric assessments. And it's interesting, when the list first came out, started to come out last year, this list was almost double in size. And as more and more information and more discussion on the targets has happened through various public workshops, this list has shrank considerably, probably much to the dismay of the sponsors developing oncology therapies. So just a couple key considerations before I wrap up my portion of the talk. Again, just because the target is not on the list, it doesn't mean that studies could, don't have to be completed. Similarly, waivers can be granted even if the targets are on the list. So, for instance, if you've got a drug where that they're for certain age groups or certain development timings that these cause considerable toxicity issues, and so you're not going to subject certain pediatric populations to serious risk or harm based on what you know about the drug from a pediatric or from a preclinical perspective. There also could be, due to patient populations, there could be difficulties in feasibility to conduct the studies, and they may look to incorporate these in other sort of adaptive or other study designs. Also, if studies are not or products are not first in class, so again, that would be situations where additional studies would not provide substantial differences in efficacy, safety, or PK. So again, these targets and inclusion of these targets on lists is really based on a totality of the evidence. And these lists are updated on a regular basis, and they have semi-annual public workshops to discuss how targets are coming on and off the list. So one of the big things, one of the big challenges that sort of came about was obviously for targets that were quite popular, and so there was a lot of challenge in prioritizing which products to study when you had targets that were being sought after by multiple sponsors. And so a lot of the FDA decision making on this would come down to the target involvement. So, is it part of multiple diseases or just a single disease? What's the mechanism of action or the profile of engaging that target? What's the potential public health impact of studies with that target versus the others? And availability of the therapy and availability of biomarkers to inform on a more robust study design. And a lot of these get worked out through collaboration through industry, key stakeholders, and regulators. So they hold these strategy forums. So they've already seen a lot of these with some of the more popular targets like ALK and checkpoint inhibitors. And so what are some of the considerations for you as a sponsor that are developing drugs that are affected by race for children? And really it is a paradigm shift. So it's really looking at it, having a proactive consideration, the integration of pediatric development within the overarching drug development plan. So if you know that you're developing a target or developing a drug within this space that is going after one of these targets, think about your formulation and your your pharmaceutical sciences aspects early on. Think about your dose and how it could be affected in the pediatric population, sort of the regimen, what other considerations you want to look at and extrapolation as you're starting to generate data. What kind of non-clinical data do you have? Do you have the appropriate animal models from both a pharmacology perspective and also from a juvenile toxicity perspective to inform on pediatric populations? Do you have the right biomarkers in place? Have you looked at the translational potential of your biomarkers and what that can inform on in your pediatric population? And finally, what kind of adaptive trial designs can you do to accelerate the development and sort of seamlessly integrate your pediatric assessments as part of your overarching development plan? And really, the key here is early partnership with stakeholders, and especially if you're looking at global development plans and really making sure that you're getting that feedback early on from both the FDA and the EMA, and making sure that everyone's in the loop and that everyone's aligned on your strategic plan. So in summary, the goal really for the Race for Children Act is to promote the access to and advancement of much needed cancer therapies for children. The focus of it is not to just increase the number of phase 1 pediatric studies, but really try to accelerate the appropriate and timely initial pediatric evaluation. Again, it's going to be dependent upon collaboration of sponsors with the regulators, and that's going to be critical for success. Again, it's integration of these pediatric assessments in the clinical development plans to generate clinically meaningful data. And so with that, I'll turn it over to Peggy, who will discuss some of the tips and tricks and operational considerations that you should consider to really develop this clinically meaningful data in peds.
Thank you, David. As David indicated, studies in children are really quite distinct from those in adults, and children are legally protected as a vulnerable population. And as such, studies involving children require special considerations. There are medical and regulatory issues that are different when children are involved, and there is the potential for impacting the child's physical and cognitive growth and development. And for all of these reasons, the pediatric oncologists and pediatric IRBs are by their very nature, they're more conservative than those conducting clinical studies in adults. The additional regulations governing research in children are, as we know, found in the Code of Federal Regulations 21, Part 50, Subpart D. And basically, these regulations state that if a study presents more than minimal risk, the risk has to be justified by the anticipated direct benefit to the child. And also that the risk-benefit profile of the study is at least as favorable as the available alternatives. So who qualifies as a child then? The regulations define children as persons who have not attained legal age for consent to research. But this is under the jurisdiction in which the research is being conducted. So it will be very critical to understand and be aware of the local and regional laws governing the age of consent, especially for global projects. Of the 4 permitted categories for research in children, most of the pediatric cancer studies are going to fall under the research involving greater than minimal risk, but presenting the prospect of direct benefit to the patient. And these studies can be conducted as long as there are provisions for obtaining the assent of the child and the permission of their parents or their legal guardians. So the consent process, as you would probably guess, is much more complex in pediatric studies than adult studies. Consent is given by proxy by either the parent, the legally appointed guardian, or a court of law if the child is a ward of the court. Ideally, you would want the IRB to allow only one parent to consent and sign the informed consent form, but it is important that both parents agree to enroll their child in the study. If this child does have a legally appointed guardian, be aware and have the sites be certain that the legal documents cover enrollment in clinical trials in addition to their standard of health care. And if they are a ward of the court, and this does happen, then they definitely are allowed to participate in clinical trials as long as the specific regulations in that section 50.56 are followed. So in addition to the parental or the legal guardian permission, what's different than adult studies is the ASSENT process. So children at certain ages will be required to give affirmative agreement that they want to participate in the research. And the ASSENT is defined that way, just as that they give their affirmative agreement to participate. The age at which they must give this ASSENT is really determined by each IRB. In general, most IRBs will start somewhere around the age of 7 years, but each IRB could be different, and it could depend on the study. Something to consider is that you'll have multiple ASSENT documents depending on the age in which your inclusion criteria are defined. So ASSENT documents will need to be written in age-appropriate language, and a separate ASSENT will be required for all of those age groups as defined by the IRB. So you'll see on the right an example of a very young age ASSENT form, which looks really quite different from a consent document that you would give an adult. And for this ASSENT form, the young child would listen to the explanation of the research of the study and then color those corresponding numbers on the document if they understand and agree. And this, you can see how different these would be than the adult consent, informed consent documents. Aside from parental permission and assent of the child, there's also the consideration in pediatric research of reconsent. A child would need to be reconsented to the study if they're still participating in it and they attain legal age. And generally that's 18 years of age in the US, but again, this would be regional jurisdiction determining that age. Important to consider is that if they are not still participating, but previously collected samples are being utilized or medical records are continuing to be reviewed, the child will still need to reconsent. So it will be important to consider and add reconsent provisions in the monitoring plan, and consent and assent and reconsent should always be part of the site training for the pediatric cancer studies. When thinking about study design in pediatric cancer trials, obviously these are rare diseases. There's a small number of patients, but as David indicated, with the development of molecularly targeted agents, these rare diseases are now really being divided into even more scarce subgroups. So consideration for a large number of sites and obviously global consideration will need to happen as we put the molecularly targeted agents in place. For the phase 1 dose finding studies, of course, the traditional, more conservative and the very safe approaches are still the 3+3 standard design, rolling 6, the algorithm-based designs. But model-based designs like the continual reassessment method can and really should be considered, and David alluded to those as the adapted designs really to try to accelerate these products in getting into the pediatric population. But the most important thing to do is to develop a really practical protocol which will set you up for successful completion of that study, and including those age subgroups as appropriate and monitoring for those age-related differences in that safety profile. Now, as opposed to adults, the bioavailability, the metabolism, the excretion of drugs is really very different as the child moves through their stages of physical development. And so your study and design and the study assessments will really have to account for those various stages of organ development. And I won't go through all of these. You can find these in the FDA E11 document, but clearly renal clearance, liver metabolism, the central nervous system is maturing during the early years, and then for the adolescents, sexual maturation, and throughout all of the pediatrics, effects on cognitive development are important. When, before actually even considering sites and investigators for your study, really very early on in the pediatric program development, you'll want to identify and engage a physician champion for your study. And this should be someone who's well respected, published, well-published, and a key opinion leader in that indication that will really help your study launch and get completed. And when considering sites, really ensure that the division director is on board with prioritizing your study, because division directors are responsible for doing that prioritization. So you really want to make sure that you're looking at sites that are going to prioritize your study for recruitment and enrollment. Try to pick sites that are referral sites for that indication, and have dedicated pediatric cancer units and staff. And when looking at principal investigators, obviously expertise in the indication, it's helpful to have national and international recognition. And if they are junior investigators, ensuring that they have training in clinical trials or they have a mentor is important. And enthusiasm for the trial will go a really long way and spread across the other sites. So recruitment and retention, when you start to get ready to enroll, something a little different from adults is that really it's a family affair. And parents obviously will need to be recruited first. And then as we discussed, children will need to give their assent, and that will be according to the IRB specified ages. Study information will need to be shared with extended family members, which may be a little different than you're used to experiencing with adults. So plan on supplying informational flyers that the parents can take home. And be aware that grandparents and significant other family members may expect to be involved in the discussions with the investigator, and actually the parents really want their input into the decision-making process. So who are the stakeholders who have influence on the parents' decision-making or the legal guardian decision-making? And there's a wide variety of people, but mainly the family relationships, the child, step-parents, grandparents, extended family members, and friends. And unique, I think, to the pediatric cancer research, the parents very much rely on the oncology staff, so the physicians, their primary nurses, social workers that they're very connected to. They do rely on their expertise and their advice, and really don't leave out the community, because the faith communities, disease advocacy groups are strong in pediatric cancer and provide support to the family. So those are other areas. And with all of that said, then the recommendations are really for recruitment are to cast a wider net for engagement than you might have considered with your adult trial. And be aware that age-appropriate flyers are going to be very important for the children, and providing materials that the parents can share will be important. Get those done up front so that those can be part of the submission package and not have to delay the IRB submissions. And a key piece to retention is really keeping the parents and the sites updated on the progress of the trial. That's been shown in research, and so that would be an important piece to keep in mind. Retention is so important in the pediatric trials. The other key consideration is family support. So these are rare diseases, and they follow along rare disease type of activities. So we know that there will be family travel to the sites. So consider concierge services. It really takes a really big burden off of the families trying to get to sites and travel. Little things like meal tickets, activities for the siblings, gaming and movie devices I found were really incredibly helpful for parents who have the siblings all with them, especially on long days where they're in the clinic. And really, as opposed to adults, you're going to have to consider all the schedules, not just the child's school and activity schedule, but the parents' work schedule and sibling schedule. So make these visits really as convenient as you possibly can. So with your pediatric study will be assessments that are unique to children, and you'll want to really critically review every line item on that schedule of assessments. I don't think I can emphasize that enough. Eliminate any research procedures as unnecessary that don't contribute to the scientific objective. That's straight from the regulations. And be aware that although the IRB is looking at the overall study as far as risk and clinical benefits. They are also looking at all of the procedures and the tests required. And if that test is more than minimal risk and providing no benefit, that won't be approved by the IRB. So the key here really is to align your procedures to the standard of care as much as possible. So, for example, in adults, tumor biopsies at the beginning and the end of treatment can be common, and adults can consent to that, but if there's no benefit to that wouldn't be approved by the IRB. So think about that when you're developing your protocol. The protocol itself is going to be really key to your operational success, and I would design it specifically for pediatrics. I see a lot of protocols that are, in my mind, they're reworked adult protocols. I wouldn't do that. I would, to make this very successful, start over, design that for specifically for the pediatric population, and involve all the stakeholders, as many as you can, and I would put in there involving the parents and the patients themselves, especially the adolescents and the young adults. There will be assessments that are unique to research in children, and you want to think about these. Obviously, physical growth and development measures will be important. Not all measures are going to be required. It's going to be dependent on your IP. But one of the things I commonly see in adult studies, height is usually only measured at the screening visit, but weight and height will need to be measured at every visit for pediatrics. This is how we are following their growth. So the growth curves on the growth chart that you see there will be important for measuring growth and development. And for very young infants, we do the head circumference, usually up to the age about 2 or 3 years old. If you're looking at skeletal growth, you're going to probably want bone ages or at least standing and sitting height, so things to consider for growth and development. And of course, the baseline assessment of organ function is going to be important for these children. One thing to seriously consider is the measures needed for sexual maturation, especially for those that are adolescents. These will mainly be involve hormonal markers of puberty, but also will involve physical signs, which are followed by the Tanner staging. Other assessments unique to children could be cognitive development, neuropsychological testing, school performance are the main measures for that, psychosocial development, such as school attendance, and then thinking about long-term effects. There may be some post-marketing surveillance that's needed, and then long-term studies may also be needed depending on your IP. A thing to consider really that might be quite different from adults is the requirement of sedation and general anesthesia, particularly for imaging procedures. Most pediatric sites will require a sedation or general anesthesia site, a general anesthesia slot for children ages 6 and under. Occasionally they'll attempt to do a 5 to 6 year old without sedation or GA, but it's generally required. They just cannot sit still for that length of procedure in, for instance, the MRI machines. Other types of sedation and general anesthesia required for procedures are bone marrow aspirates and biopsies and lumbar punctures. So important really to put those procedures along with their standard of care as much as possible. Other types of considerations, drug formulation is so important. David touched on this, but very early, think about age-appropriate dosage forms, suspensions, liquids, chewable tablets for injectables, small volume vials, and as much as possible, long-acting formulations. Adherence is so much easier if it's given, for instance, once a day versus 4 times a day. And dosing for pediatrics is usually in milligrams per kilogram of body weight, but some of these drugs with narrow therapeutic indexes will be based on body surface area, milligrams per meter squared. But all of these things are important: the taste, the stability, and then for pediatrics, excipients needs to be thought about because adverse reactions we know have been observed in very young children, neonates and preterms that have not been observed in the adult population. Adherence to the actual medication is so important in pediatrics. most of the oral medication for adults tend to be tablets. And if your protocol involves children about the age of 8 years and younger, can they swallow the tablets? Is that going to be successful? Because I will tell you what will happen is you'll have the parents trying to crush that tablet, mix it with fruit juice, mix it with food or milk. So consider all these types of things. If the tablet is the only formulation, can they do that? It's better to have a liquid or suspension, but even with liquid and suspension, what's the volume? If you have inclusion criteria of age 2 and above, and you're asking them to take 4 to 8 ounces at one time, that's going to be problematic. And then just be aware that what will happen is if this is difficult for the child to take, if it's a formulation that's difficult, they will vomit, they'll spit, they'll hide the medication and refuse it. I would highly encourage you to consider, especially in the pediatric cancer population, can your drug be given through a gastric tube or the G-tube or the nasogastric tube, because a lot of the children will have those for nutrition. And then thinking about something that you don't have to think about with adults is, do you need to give this at school? If so, there'll be some additional paperwork that will need to be processed for school administration of investigational products. And then you can't have a talk about pediatrics without talking about pediatric blood volumes, because the IRBs will have to review the amount of blood that you're taking, and it will be limited to a percentage of the child's estimated total blood volume, both at any single one point in time and cumulative volumes over a defined time period. And that's generally 30 days, but it's really very IRB specific. And so there are World Health Organization guidelines that's referenced there. They're widely accepted, but they're not going to represent restrictions across all IRBs or ethic committees. So you have to really lay that out in your protocol and in your informed consent, what the amount of blood you're taking, and be aware that they will want to know the research amount plus the clinical volume. Other considerations would be the number of venipunctures or sticks that's acceptable, numbers of entries into central lines, because we know that multiple entries increase the risk of infection, and that you're minimizing discomfort and pain and emotional distress by providing anesthetics or topical kinds of medications that help with these. So when looking at your study, strategies that you can use are to choose pharmacokinetic techniques that really minimize the number of samples, utilize assays that require less blood, and then I cannot emphasize this enough, but in your protocol, and maybe even as a tool for the site that they can carry, prioritize which samples should be drawn first. So what is your top priority for research samples? Make a list. If they are getting to the point where they can't draw any more blood, make sure that you get that you've prioritized and you get the top things that you want for that study, and then really specify the minimum volumes that you need. Urine is it's interesting. I spent my career in pediatrics, so this was never a thought to me. But obviously, with adults, if you're collecting urine, it's pretty easy. With children, it's just not that simple. We have a lot of ways to get urine, one of them being the PD bag that you see there. So bag collection of urine, consider is cotton balls in the diaper appropriate where you squeeze out the urine. Clean midstream catches are really challenging even for the toilet-trained children. And 24-hour urine collections, we can do them, but it's difficult, and so really think twice about requiring that. Other challenging issues that you might come across are just the sensitive type of information, particularly for adolescents, pregnancy testing in young females, any kind of contraceptive requirements parents may balk at. Think about those carefully. And then your consent and ASSENT forms need to describe plans for disclosure or non-disclosure to parents, legal authorities, and really the patients themselves for any really sensitive things that you will see there, like sexual activities, sexually transmitted disease, and HIV status. And if your product is affecting fertility, you'll want to consider fertility preservation in your protocol. So in summary, children are a vulnerable population. They will have unique regulatory protections. Write your protocol specifically for children. Involve your pediatric cancer experts and your other stakeholders, and I would include parents and patients in that. We get a lot of good advice from our parents. Align your protocol assessments with the standard of care as much as possible. It's really going to help keep your budget down, and it will help get it through the IRB. Address potential effects on growth and development, both physical, cognitive growth and development. And early on, consider appropriate drug formulations and the impact that that formulation will have on administration and adherence. And then choose experienced pediatric cancer sites and investigators, and really consider the family in the recruitment and retention strategy, the whole family, and not just the patient or the parents themselves. And with that, that's the end of the presentation, and I'll hand it back to Candace. And thank you very much.
Well, thank you very much for that very insightful presentation. At this time, I'd like to invite our audience members to continue submitting their questions or comments right now using the questions window for the Q&A portion of this webinar. And to take us through the Q&A, I'd like to hand it back over to our speakers, David Horton and Peggy Kaiser.
Okay, thank you very much, Candace. So I think one of the first questions that I see is how should companies initiate early interactions with the FDA? So one of the clear pieces of advice the FDA have through all their initiatives around the Race for Children Act has been to engage the FDA early. Certainly you can engage and ask questions regarding your pediatric development plan all through your development as part of your standard development meetings. However, sponsors have the ability to specifically request a meeting with the oncology CAV, then the pediatric oncology program, to discuss their PSP or to discuss study design considerations for programs with relevant molecular targets. And so, again, what that meeting would look like would be, let's discuss, the relevance of the target. Is there options for waivers? Is there options for partial waivers, for deferrals? What are the FDA's expectations around assessment here? And really, what you would do to engage this type of meeting would be you would send this to the review division with your cover letter specifically stating that you're looking for a meeting for preparation of IPSP under FDARA, and they'll schedule that meeting within 30 days. So it's a pretty fast clock. But with that, they really want you to submit your briefing package to alongside your request for a meeting.
Okay, and there's a question that is, how do you involve parents and patients in the development of the protocol? Well, I think the best way to involve parents is really to include them on your advisory board. And also, this should be older patients as well as young adults, adolescents and young adults. You can solicit the help of your physician champion or the PIs to identify appropriate patients or parent candidates, and of course they'll need probably to sign the confidentiality agreements just like any other member. But patient and parents are really very willing to review the protocols and offer really good advice, so I highly recommend it. Another way that I'm familiar with is conducting like one-time focus groups, and I think you could get the participants in the same way by really asking for help from or soliciting help from your physician champions and the other key opinion leaders. Parents are very willing to do this, so I highly recommend it.
Okay. Another question came in terms of what is the FDA's expectation around non-clinical data to support the pediatric assessments and the PSPs? So this is a great question. So the FDA sort of looks at this as breaking down sort of pharmacology and toxicology. So from a pharmacology perspective, what is the data telling you with respect to, your pediatric model? So, how well would that data in that model and how good is that pediatric model to inform on potential efficacy in the pediatric population in the clinic? And so, here, this is where translational biomarkers come in, biomarkers that would really inform on potential PK-PD relationships, monitorability of safety effects, which I'll get into in a second, sort of clinical efficacy. Is there anything unique to children that would make this biomarker not relevant, make a biomarker that you observe in adults not relevant for children? And then from a safety toxicity perspective, I think it's really what information do you have from your tox studies to support pediatric dosing. I think key here is your target organ. So do you have toxicity or do you have effects in your developing organs which would not allow you to predict what is going to happen when you transition from adults to pediatric population. So as Peggy mentioned, obviously the organs of interest here would be the brain, the central nervous system, are there effects there? Skeletal is a big one for, development, and obviously the reproductive organs. So, and those really would try to inform on potential development issues and ones that you'd really want to understand further. And then finally, sort of how, the animals that you were used for your tox study, how can you extrapolate that out to support the age in the clinic? So it could be that you've used adolescent animals and you could already have existing support for testing at a lower patient population.
Okay, I think I can take this one. How much blood does an IRB allow to be drawn from children at one time? Well, this is really going to be specific to each IRB, but in general, I've seen anywhere from about 1 to 5% of the child's estimated total blood volume can be pretty safely drawn at one time. If you have like a 6-month-old child and their estimated blood volume is 630 milliliters, that's going to be around, 6 mils at the minimum. So you can see these are small volumes of blood for the youngest children. As you get up through the adolescence, it's a little more, they are able to tolerate a little more blood drawn. But each IRB will be different, and you should really get that IRB's definition.
Okay. I think in the interest of time, we'll hand this back over to Candace, but we certainly appreciate everyone's involvement in the webinar and their insightful questions.
Well, thank you very much for those answers. We've now reached the end of the question and answer portion of this webinar. If we were unable to attend to your questions during the Q&A, the team at Medpace may try to follow up with you. If you have further questions, please direct them to the contact information showing on your screen. We also encourage you to register for Medpace's upcoming webinar, Challenges and Considerations in Clinical Development of Targeted Therapies, AML as a Case Study. I've included this link in the chat box. At this time, I'd like to thank everyone for participating in today's conference. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve our webinars. Now please join us in thanking our speakers, Dr. David Horton and Peggy Kaiser, and feel free to share the recorded version of this webinar when it becomes available using the link in the chat box. We hope you found this conference informative. Have a great day, everyone.